Yes, so maybe you asked the question, oh, what is this? This is a title with a question mark, like EVB scanning database, what should that be? And actually you're right, because it's the first time for me to not give a talk about my research, but instead about something completely different. And probably in this talk, I raise more questions than I answer questions, but we still thought it is a very wise idea to put it right now here, because I think this is the perfect moment to do that. So where did everything start? Actually, I would like to tell you something about the European Virus Bioinformatics Center, which we have founded something like in 2017. And the idea was actually back then to bring the bioinformaticians and virologists together to exchange ideas, to specifically design tools which the virologists need, because all the other tools for bacteria may not work out for viruses. And yeah, also start a lot of scientific cooperations. And instead of everybody doing a very small part in his group, we really try to do something together and really drive forward the field of virus bioinformatics. And yeah, meanwhile, we have quite a lot of members, and some of them you may know. So I just put some of them here, which have also been here during the conference and the audience. And what is actually this European Virus Bioinformatics Center doing at the moment? Actually, it's not only for Europeans, so everybody could participate. Don't ask, this is a longer story why it's called European Virus Bioinformatics Center. However, so what we are doing is we try to test tools for the virologists. We try to sort them, to filter them, and so on, and have a place where all the tools which are available are kind of collected, at least their links and maybe a kind of description. So you may look for that. Or we have also once per month, our Virus in Silico EVBC lecture series, and quite a lot of people also from the audience were already talking there. And the next one will be Bas Duttel, probably you know him, Mr. Cressfate, and he will talk on the 25th of October. You are heartily invited to join, of course. We also try to set up a calendar where we have all the important virus workshops, conferences, but also bioinformatics workshops, which contain viruses as a part. And yeah, you can see here in the middle, our nice workshop is also here right now. And beside these three things, we also try to gain larger projects, and we were also successful because now it's including 45 people. It's an EU project, actually, which tries to understand the virus host interactions, and this is half virologists, half bioinformaticians, really driving the field forward. And besides trying to foster such projects, we also try to spread all our news via our newsletter, which you, of course, don't get by snail mail, but by email, of course. And you can become a member if you want. And yeah, it costs nothing, and you profit, hopefully, from all these services. And additionally, once per year, we have the International Virus Bioinformatics meeting. So here you see one picture from Glasgow with this nice building, from Utrecht, and the last one was actually online. And it's actually a kind of family coming together. And when we came the first time together, I was asking, hey guys, what is the most important thing which we actually need? What the virologists need from the bioinformaticians? What tool we should develop? What is most important? And then it kind of immediately, all people said, well, we need a database. We need to store our data. And I was saying, hey, wait, why you need a database? You can just take the common database and you can just put your virus in there. What's the problem? And then the virologists had wrinkles on their forehead and were just saying, did you try to do that before? I mean, if I try to put my virus somehow inside the database, they ask me first question, what is my chromosome? But we don't have a chromosome. What is that? And yeah, then I understood, problem detected, okay, all right. But it's not the only problem which actually occurs, because viruses are so specific and so diverse, and they don't fit into normal something like databases. And therefore, yeah, we were thinking, what can we actually do? And the next meeting about viruses and bioinformatics in general, and also discussion about what a user may want from a database, will be actually in Spain. So if you are eager to come, you're invited to come also to Spain and discuss with us. So what happened when we discussed, do we need a database? There are so many great databases already out there. We heard a lot today, also in the last days, some pipelines. And I just discovered that they do a good job, but actually there is way more to be done. And the question is, what has to be done, actually? And therefore, in the last meetings, we already generated a kind of to-do list. in case, for some reason, one day we have money and we have also the resources to implement something, we could go maybe about the following things. And this is actually which came out. So there is something like virus information in general. You want to have the genome sequence, annotation, transcription, products, protein sequences. You want also some information about morphology, some pictures about the virus, maybe some marker genes, you want to know about modifications and so on. But you also want to know a lot about metadata, like, okay, where has it been sampled and so on. We heard a lot about that already today, so you're aware of that. But also, what about downstream analysis? We do need alignments. We had the topic several times today. We have so many sequences. We cannot process all of them. We cannot make an alignment out of all of them. So how to select the good viruses, which represent actually what we need? And a lot of questions related to that, which is a kind of unanswered for now. And of course, you want to have links, and the user want to have also maybe a workspace where to work. Nobody can see it, but maybe it should be in the cloud. He has no space at his own computer and things like this. And there are a lot of such questions. And at the moment, we were thinking, okay, this is a wonderful, great list, but nice to have the list, but there's nobody who can implement it because we are missing the resources for that. That is really a problem. And it came really luckily to a point that we got quite some funding. So it's actually like not only one person for 3 years and then the database dies again because nobody is really maintaining it anymore. But instead, we now gathered money for 5 people. And for 2 times 5 years, so maybe up to 10 years, we can run now a kind of database. Now, the thing is, we don't want to compete with other databases at all. So I think this is a way too small community to do something, but even if it would be larger, this is not really our aim. So instead, we really would like to embrace what is existing already and try to help with the resources we have now. And so I'm thinking about eukaryotic viruses. So there are some specific databases for influenza and so on. But there's also the Viper. You may have heard about it. We heard a lot about phage databases today. And yeah, we would like to contribute to complement that actually and integrate maybe some of the tools and so on and make all the communication. That's why now it's time to put aside your popcorn and do something, because Joanne is so nice and is posting now a link into the chat, which you may use. So think about, it's October now, Christmas is coming soon, it's December soonish. So think about what would be your dream, which you want to have from Santa Claus on your wish list, what a database should actually do. So you may click the link and just write, oh, I always wanted to have this and that in the database. Now it's your time point to put it onto the list. And for all the people out there, which are the real database heroes, so maybe you run a small database and you would like to see that integrated, or you want some help to make it a little bit larger or whatever, please contact us and let's put some efforts together. So this is actually the idea. So let's see what actually can be done. And this funding comes actually from a resource called NFDI for Microbiota. And I will just briefly give you an introduction in what does it actually mean. So the NFDI for Microbiota is a national infrastructure which takes care of existing databases, also bacterial databases. And here we create the virus database, and it has a very strict policy and a lot of things which are actually included. Like part of this consortium is actually to have a lot of training and education, not only for the users, for the virologists themselves, but also for the people maybe running smaller databases, how to integrate the data structure, synchronizing, and so on. But also have a lot of support, maybe by data stewards, data curators, also developers, of course, and coordinators, and bring that together and really reach out to the community at that point. Then we follow a lot of policies and standards. So we follow, of course, the FAIR principle. We have some legal issues which we solve in this consortia, of course. And of course, we want to manage the data itself. And then we also try to set up standards. I mean, it is very hard sometimes to compare actually the data set because we don't follow the standards. Is there some standard so far? I think it's also an open question, right? So we try to, maybe give a guideline if people would like to contribute with their data data in a very comparative way, maybe with some experimental procedures, which may be some kind of standard, but also workflows like how to sequence and things like that, that you can make your data set also comparable to others. So for that, you need to know what is the kind of standard. And this is also something which is generated here. And finally, of course, the research community, most important part, should have access to all these things in the background via some web portal. However, the idea is also for all the bioinformaticians out there, you can also download and some part of the database or a larger part and also work on it on your terminal. So having said all these things, you may think, okay, this is quite a huge thing. And I'm also a little bit scared. I mean, it's a lot of resources and there's a lot to do. We need to sort out which should be done first. So if you have some hints, just let us know. That would be great. And you see already, this is a lot of puzzle pieces should come together to make really a huge resource and a huge database out of it, which everybody uses. And now I would like to introduce you to some tiny, small puzzle pieces. And these puzzle pieces, I will not mention the standard things, which have been already mentioned many times today. I would like to show you a little bit what people in EVBC and also outside EVBC are working at the moment to make the life of virologists a little bit easier in the future. One part is visualization. So virologists raised the thing, hey, I want to see my virus. Please make it possible that we can see our viruses. It is possible already, but it's a lot of effort and not so easy. Usually, you know that experimentally with a lot of ER and a lot of things. Now, there's already quite a lot of, and also due to the lack of SARS-CoV-2, a lot of things going on. Like, you can already distinguish now viruses from pseudoviruses by several techniques. There are several groups working on that, and the publications are on the way. Another thing is actually that maybe this doesn't count too much for phages, but it's a little bit different, little bit similar, sorry, that you can actually, also try to understand tropism via, pictures you can generate a bit better. Or maybe here's some example of one group which is pretty exciting. They are using the technique TERS. TERS stands for Tip Enhanced Raman Spectroscopy. And what happens there is actually you have a very tiny tip, and that goes really down to a nanometer scale. And then you have a virus, and then it can actually kind of screen the surface of the virus. So, and it doesn't screen it with taking picture by picture like with a camera, but instead it uses Raman spectroscopy on such a nanoscale level, which means that you can really see atom by atom like that via Raman spectroscopy. And therefore you can actually really say, what is the surface protein? What changes have been done there? What modifications do we have there? So that is on the way. The technique is published already, but for viruses, it's on the way to be established right now. Maybe something else to talk about is actually the kind of data structure. And this is very computational now, so I feel very well there, but maybe it's too much for you. So I would like to introduce you to some very easy general concept people are working at the moment. So just imagine you have a virus, and you have it as a FASTA format, and you store it in a database. And then you have another virus. You have another FASTA file, and you also store it in a database. And actually the difference, maybe they are just two haplotypes coming from the same quasi-species or whatever. They are just differing in one nucleotide. Then you may think, okay, that's fine for two viruses, but if I have a lot of them, how can I save all of them? And that's why people think about how to store the data in a different way. One approach is actually something which comes from the field of pangenomics, where you actually store each nucleotide as a node in a graph. And you connect it like that. And now if you have a second virus which just differ in one nucleotide, then you may just tell the graph, okay, after A and T, you can also go for G and then back to the genome as it was already. And you see already that in terms of storage, this is way smaller. Well, that sounds fantastic, but it's not that easy, as always. So for example, you can also imagine the genome goes longer and maybe there's another nucleotide exchange. But what people are actually interested in is, do these two exchanges come together or not? So are they linked? And if yes, how to save it and store it in a database? And that is becoming pretty difficult right now. So that's why it's actually an open bioinformatical problem at the moment, how to save the structure in a very sensible way. And so people work on that as well. And maybe you see a little bit what happens in the back. But maybe I can also introduce you into another topic which happens, and I will take the example of a virus-host relation. So let's assume people are already able to make a phylogenetic tree of viruses. So here you have a lot of different phages. I actually just learned a few days ago there are also phages having really cool shapes. And let's say you have a phylogenetic tree, and you also have a phylogenetic tree of the host. Then actually, bioinformatically, we are able to find the kind of correlation if this virus infects actually that host. And already that is computationally interesting, but that is easy, but that's not reality. Reality looks way different. Reality is, of course, that you have some very strange interaction. And then the computational people, they say, maybe we need to reorder some of these phylogenetic trees, or maybe we need to do something else to really have this nice correlation. And it comes even worse, because a virus does not need necessarily only one host, but maybe it has even several hosts. And how to bring that together? And we think having such a powerful database, maybe it is possible to take different features, different attributes than the virus genome and its nucleotide comparison and the host genome and its nucleotide comparison. Maybe we can take actually something like, let's say, the folding of a surface protein, which determines actually where to go to which host. And maybe we can cluster viruses and also hosts in a different way and have a nicer correlation. Things like this would be a dream to have out of a database. Now, I also would like to introduce you into some more computational concepts from our group. For example, the question is, how do I actually, I said it already, select a representative virus out of a huge crowd of viruses? And actually, there are several things or possibilities at the moment, and we just developed a tool which might be useful. So it's not new to have a lot of input sequences and to make something called a k-mer profile. In this case, you see the sequence is only 7 nucleotides long, so it's a 7-mer profile. And you can just say how often you have it per genome. So each dot is one genome, and you have a feature vector, more or less. And then each of these lines actually is one dimension, and you have a very high dimension, which you can actually very hard process. That's why we try to find a generic way to reduce these dimensions. We use something similar to UMAP, and then we can actually cluster them, and we try to find something like a centroid sequence, which represents this cluster, hopefully most powerful, and really taking all these centroid sequences together may serve as a good input for something like an alignment or for classification of viruses and so on. Actually, we tried that here. This is not phages, this is filoviruses here. And you can see this is the phylogenetic tree, and you see blue dots here and there, which actually are the centroid sequences which represent the sequence. If we zoom in, we complement it also at the end by ICTV representative viruses, which the virologists actually use in the lab, to our alignment. And then we ended up making this kind of cluster, adding the very important virus sequences from virologists, having a tree, making alignments, and we extend our alignments even in a way that we don't use only sequence level, but also intramolecular interactions and secondary structures to gain final alignment. And I think I would just like to show you maybe one more example. Here's also some secondary structure of multiple sequence alignment. This is the 5' UTR of coronaviruses. We were working on that way before we had the outbreak, actually, and it was just on the right spot that we had these data already available. So this was work from Markus Fricke. Yes, and with that, I would like to show you maybe a last thing. We are also trying to generate another puzzle piece, which hopefully at the end can be part of this huge database, where we actually try to tackle another layer of information. As you know, DNA has some modification, sometimes like acetylation or methylation. But did you know that RNAs also have modification? It's actually something like 140. And currently, we can have something like a direct RNA sequencing. We cooperate very closely with the Oxford Nanopore Technology Company, with the MinION Nanopore. And we try to read, or they call it base call, the sequence out of such a current track. And actually, if you have a modified nucleotide, you can base call and identify such a modification. However, at the moment, we do not have the bioinformatical tools for that because this is usually a neural network which has to be trained. And that's why we at the moment do a lot of different in vitro transcriptions, incorporating several modifications and try to learn how these modifications look in such a nice current plot, and then, or they call it squiggle, and then trace it back to the modification. And hopefully that can be also of importance in the future and be part of a database. So let's see if that happens or not. And with that, I would like to finish. I hope I gave you some puzzle piece ideas, but there is so much, and I would be so happy if you could contribute to our idea which we try to bring together. And with that, I want to thank my group. I thank you for your attention, and I hope you had a wonderful conference.
Fantastic. Thank you so much. That was a wonderful talk and also highly entertaining in addition to being scientifically fabulous. So we have a lot of questions for the panel, and I think I'm going to start. Please feel free to type questions specifically for Manja now if you have any, but I have a quick one, which is that this looked like, you know, just an incredible potential resource, right? Like lots of information that you can be pulling from disparate sources. And I was wondering if you have a feel for, you know, sort of what combination of examples you've shown us so far you think would be most applicable to phages that are already really well curated in public databases versus, for example, I have, you know, my pet viral sequence from a virome from soil that, you know, doesn't match a whole heck of a lot. What's sort of the use case for most of what you're developing?
Well, I think the question needs to be answered actually in two different ways. One's for the phages and one's for non-phages, because this is quite different also what is needed. Talking about maybe viruses which are pathogens, there is this resource which is the Viper database, which actually holds all the human pathogen viruses, which is great. And it also gives a lot of possibilities using tools and a lot of other things, which actually helps, and people use it very much. So this is great. But it needs to be extended. And I think the biggest problem, and that counts even more for the phage community, is how to make the people actually upload all their data in this meaningful and sensible way. And I think this is something, it needs actually quite a lot of talk. I mean, today already Simon said it in his talk, and also Rob said it in his talk. If you have ideas how to improve it, please tell us. But this is really the most important part, because as a computer scientist, we can just guess what you have problems with and what you need. And yeah, actually it would be great if you give in whatever way some feedback. And I think like that we are able to really make the community better, making them using databases and also making it possible to really work with it, because we are just at the start doing that. Yeah. So I don't know if that really answers your question, but I think this is so important, so I needed to emphasize it once more.
No, that's fantastic. And I think that's absolutely right. I mean, the answer is we as a field need the database or the combination of databases, and we need to have the discussion as a field to determine what it is that we need moving forward. And so we have the right people in the room to start that discussion right now. So I'm going to pull some of the questions. We're going to shift over to our panel discussion, of course, at this point. And